Hi everyone, and welcome to the second installment of our series on getting started with standards and reference materials for use in microbiome analysis. This section is going to cover examples of references and how to use them, or as I like to call it, microbial tools of the trade. So to start off, I'll show the ZymoBIOMICS mock microbial community standard, which is available as both whole cell and genomic DNA extract. This is a microbiome standard. It is a mock community of well-defined composition. It's made of very kind of standard microbes that would be available from an organization such as USDA for testing purposes. And while it wasn't built to mimic any specific microbial community, it was built to help analyze a few different properties of pipelines, including things such as lysis or lytic challenges. And one of the ways that this was accomplished was by having a known relative composition mixture containing gram-negative microbes such as Pseudomonas, E. coli, and Salmonella that tend to be very easy to lyse, gram-positive microbes such as Lactobacillus, Enterococcus, Staph, Listeria, and B. subtilis that tend to be much more difficult to lyse, and two yeasts, including Saccharomyces and Cryptococcus, and these tend to be both extremely difficult to lyse but also very different cell size, which can affect lysis itself. The abundances of these, and knowing that there's different lysis difficulties for these microbes, it then becomes relatively easy to start picking up things such as biases caused by lysis. There's also a wide range of GC content, and while lysis is very unlikely to be affected by GC content, library prep for sequencing can be very strongly affected by GC content, and that can create significant biases during microbiome analysis. So being able to detect that through a standard is also extremely useful. I showed in my previous section that there's a family of ZymoBIOMICS microbial standards, and this particular standard fits into the family of mock microbial communities, both whole cell and extracted genomic DNA samples. In particular, they sit right here with the ZymoBIOMICS microbial community DNA and whole cell. And it's very useful to have both of these available for reasons I'll discuss in a moment. So both of these standards, the mock microbial community gDNA extract as well as the whole cell standard, provides us with a known input reference material with a known truth value. So because this is made in a laboratory, we know exactly what went into it, at least in what relative abundance is. And that gives us something to compare to, or an absolute known truth that we can say something is more or less accurate against. Obviously, this is handmade, so it's going to have also a manufacturing tolerance, and that can be factored into these analyses. This is available as whole cell and genomic DNA extracts, and this can help differentiate between issues that are happening during lysis and issues that are happening during the library prep and sequencing process. And I'll talk in much more detail about that in a later section about where this can be put in, how it can be analyzed, and how it can be used to identify where the deficiencies might be entering your pipeline, causing biases or inaccuracy. There's already an analysis pipeline for this established. It comes in the form of MicScore, which is a pre-built bioinformatics Docker or code base that can just be easily downloaded. It can also be run through a web interface provided by Zymo that will generate a report showing accuracy, as well as showing some figures that can help you identify specific patterns of bias that are known to potentially exist using this microbial standard. Another very different microbial standard would be the ZymoBIOMICS Fecal Reference with TruMatrix technology. And that's actually not only described in that box, but also the tube next to it, which you'll notice has a brown color because it's made from actual fecal samples. This reference material is a real-life representation. So this is a microbiome quality control made from real human fecal matter. That has the advantages that it's going to have the real matrix you'd expect to deal with, so any challenges this matrix might pose are going to be seen when dealing with the standard. This is what I would classify, using the terminology I presented in the last section, as a complex standard. This standard is going to be consistent, so even though we don't have a known truth value for it, there's about 2 million aliquots of it available, and because of the way it was produced, one can expect that the first aliquot and the 2 millionth aliquot should be as identical as possible. It's also well characterized, and while that's not necessarily part of being a standard, it does help. There's been a lot of sequencing done on this, a lot of testing, and as a consequence of that, there are a lot of references to use here, even though this isn't an absolutely known input with a known truth value. There have been a lot of things done on this, such as long-read sequencing, metatranscriptomic analysis, and so on, that can give you something to compare to, even if you don't really have experience using this or haven't used to compare to anything else. As I mentioned before, this is a real-life representation of fecal samples. So over 300 species have already been identified from here, and I think actually even after this slide was made, that number has gone up significantly. I think we're now well over 400. So this ZymoBIOMICS fecal reference with TruMatrix technology, it, as I said before, is a complex standard. It's naturally derived, it's multiple samples from multiple donors, and while that deprives it of having kind of a known truth value, so there really isn't a way to say somebody is more or less accurate relative to a known input, it does have the advantage that it's designed to mimic a real sample. And because this is multiple donors and multiple samples per donor, not only would I say that this sample is expected to recapitulate the complexity of a typical fecal sample, I'd actually say that this sample will exceed any complexity you would expect from a normal fecal sample. So if your bioinformatics or other pipeline can handle the complexity of this standard, you can rest pretty well assured that it can handle the complexity of a typical fecal sample. This sample should be coming in unlicensed and highly reproducible. Now, there will obviously be some lysis during the processing and preservation, but there was no specific lysis step carried out on this. The processing was only focused on preserving and aliquoting the sample and making sure that aliquot 1 and aliquot 2 million were as identical as possible. So about 2 million aliquots, as I said a few times, are available, and you would expect each of these aliquots to behave the same given the same treatment. There are quite a few options for analysis. Again, this is a real sample, so it can go through just about any type of analytic pipeline you would normally put it through. This sample is going to give you a lot of options for consistency testing. So in this case, we see here 6 different samples that look nearly identical. The way that these were generated, we have 6 separate runs. We used 100 microliters of the standard reference material per run. They were all lysed using the Vortex Genie with horizontal adapter, which is my personal favorite lysis method, using the Zymo beads. Max speed for 40 minutes, and then they were subjected to 16S sequencing with V3, V4 as the target region. And as we can see here, all of these yielded almost identical results, indicating a high degree of similarity run to run. And I'll talk more about doing this kind of analysis later, because this is also critical for testing a pipeline being established or evaluating a pipeline that has already been established. This can also be used to compare site to site. So looking site to site, we see here most sites are pretty similar. It looks like LabCube might have an overestimation of Bacteroides relative to the others and maybe an underestimation of firmicutes. If I was running a multi-site study, I probably would be a little bit concerned about that, and I might try and go check in on what LabQ is doing that might be giving them slightly different results. You know, it could be an underperforming piece of equipment, but this would give me something to go on and even a pattern to potentially say I know what might generate this pattern of inconsistency. If you're someone who's just starting with this and want something to compare to, I have very good news for you. Zymo has actually been maintaining a database of results submitted by both ourselves and other entities for what they got from these samples. So it's going to be different lysis methods, different extraction methods, and different sequencing methods. And even if you haven't handled this sample before and you're just getting started, you should be able to find something in here that's similar to what you've done or even the same as what you've done. And if you haven't, not to worry, because we're still actively collecting. And we want you to contribute your reads as well, so that people down the road can benefit from the efforts you've carried out. Now moving into the last category of microbial standards that I wanted to talk about, we have our spike-ins. And the spike-ins are going to be different in terms of how they're used during the process, because spike-ins don't go in as their own standard reference material or their own sample. They actually are designed to be added to an already collected sample, typically prior to the lysis step. These are available in high and low microbial loads, so you do need to do a little bit of guesswork here, but you need to select the concentration that's most appropriate for your sample, and that's because that's going to decide what the concentrations are of different microbes in the standard. Obviously, you don't want to use an extremely high load if your microbial sample is extremely sparse. That could cause you to only end up reading, or almost entirely end up reading, the spike-in and miss some of the depth you'd want from your own sample. These are absolutely quantified, so these are created with an absolutely known cell count per unit of volume, And I'll talk more about this later, but this gives you a little bit of ability to have a microbial yardstick to compare to. These are what I would consider to be unusual but well-characterized microbes. So we have reference genomes on these microbes, and these microbes are things you would not find in most environments. You would definitely not find them in a human, especially you would not find them in a human stool sample. So these again they're extremophiles. In particular, we have 2 halophiles, and if you use the low microbial load, you're also going to get a radiotolerant microbe, but you would not expect to find any of those in a typical human sample from pretty much anywhere. So because these are absolutely quantified, again, it gives us some interesting things we can do. Both microbes, so for the high microbial load standard, because both microbes are present in known absolute counts. First, we can look at the ratio between the two. So Imticella is an easy to lyse gram negative. Allobacillus is a tough to lyse gram positive. And so that immediately gives us the ability to detect differences in what we observe versus the expected ratio. And if we see significant changes, that already can raise a red flag about maybe a poor lysis event in a sample or some other potential issues. But it also gives them the ability to function as a microbial yardstick. Because if these go in the same amounts in 2 different samples, and because they're easy to identify compared to regular samples, again, these are going to be very unusual, very divergent microbes from what you'd find in a typical, say, human sample, we can pretty easily compare how many reads we're getting from spike-in versus how many reads we're getting from our biological origin sample, or I guess I should say our sort of wild origin sample, and use the spike-in as a microbial yardstick. I'll talk a lot more about that later on during this series. Because these samples, again, are easy to differentiate, they're also easy to bioinformatically isolate from your real sample and run them through separate pipelines to get somewhat different properties. So in this case, what we're showing here is we start off with our whole sample. We bioinformatically isolate out Allobacillus halotolerance and Imticella halotolerance. We then calculate the absolute abundances of other microbes if we are so able to. That obviously requires a little bit of prior information as well. But we also just gain the ability to compare the abundance of other microbes in total to our spike-ins, and that will actually give us a very good idea for just the overall microbial richness of the sample. And that brings us back to typical starting questions when trying to employ a microbial reference material, and that's what we're going to cover in the next section. So I will catch you next time for section 3, where we will talk about the first 3 questions, which are the kind of questions you want to ask when you're just starting to establish your microbiome pipeline, or maybe thinking about doing a refresh. Thank you, and I'll see you in the next section.